Kia ora koutou, and welcome to our digital event. It's great to have you join us tonight. I'm going to start off by reading a quote that hangs on my wall, which really gives the essence of what research is all about. Ahara takatoa e te toa takatahi, engari he te toa takatini. My strength is not as an individual, but as... So it gives me great pleasure tonight to introduce our speaker, Associate Professor Caroline Beck from the University of Otago in Dunedin. Has started her own lab right back in 2004. She was the Director of Genetics at the University of Otago from 2017 to 2020, and she's been a two-time recipient of a Neurological Foundation project grant. The first one was in 2021. The second one she was awarded in 2023, and that's currently ongoing, due to finish in 2026. So welcome, Caroline. It's great to have you joining us tonight.
Kia ora, Sarah, and thank you very much. I am just struggling to get my video on. There we go. There I am. Hi.
Hi, thanks so much for joining us. And before we start your talk tonight, I thought it would be really interesting to hear about your journey to becoming an epilepsy researcher.
Yeah, so I'll try and keep a very long story short, but I'm a developmental biologist by trade. So it means that I've spent most of my career, about 30 years now, studying how embryos develop from a single cell to a multicellular complex organism with tissues and organs and systems that all function beautifully, including, of course, our wonderful brains. And so three things really have pulled me away from looking at things like limb development and tail regeneration into looking at epilepsy and what I can do to try and help with that. And those three things are basically two things that are out of my control and one thing that is very much in my control, and that is my middle son, Owen. He was born 21 years ago and was diagnosed with developmental epilepsy. So he started to have seizures very early on in his life. So that's really triggered my interest in learning as much as I can about epilepsy. The second thing really is that when my son was born in 2003, we didn't really know much about human genomes. We only had one, the human reference genome, and now we have thousands and thousands of them, including very many from children like my son who have an epilepsy condition or some other disorder. So we know really what the causes are of these disorders. And that brings them back to this developmental origin. So now we have the cause, we can perhaps go back and try to understand how the change in the genetic sequence that occurs in these children changes the way that their brains develop and predisposes them to epilepsy. And I guess the final thing is the development of CRISPR-Cas editing tools, which has just made it very easy for people like me to model genetic epilepsies and other conditions using our model organisms. So that's it in a really concise manner.
Well, I look really forward to hearing your story tonight about how your personal experience combined with the expertise and the latest technology is doing some really interesting work. So I invite you to share your talk with us now. Thanks so much, Caroline.
Okay, so hopefully you can see my screen now. And
Yep,
thank you so much, everybody, for joining me in this quite short talk, I think, hopefully, about the genetic mysteries of early onset epilepsy disorders. And a huge thank you to the Neurological Foundation and Sarah for hosting this. As Sarah said, I'm from the University of Otago. I've been teaching here for 20 years. I mainly teach genetic students. I also teach first-year health sciences students. Some of you may even have been taught by me. It's possible. And so, yeah, just want to talk today about my research that is funded by the Neurological Foundation on early onset epilepsy. So I thought I'd broaden it out to start with and just look at what is epilepsy, because not everybody has the same image in their head when they think about epilepsy. So first of all, it's reasonably common. About 1 in 26 people will actually develop epilepsy at some time in their life. And within a given group of people, about 1 in 100 of those will have epilepsy. So if you walk into a room of 200 people, 2 of them may have epilepsy. You may not know that because that may be very well controlled, but they nevertheless, they have epilepsy. So it's reasonably common. And in fact, in children, it's the most common neurological disorder. I think overall it's about 4th ranked, but in children, it is really, really common. It's the most common neurological disorder that's diagnosed. Of course, epilepsy can occur at any age, but I'm mainly interested in the children at the moment. And of course, we associate epilepsy with seizures, and people have very strong images in their heads probably when they think about seizures. But seizures are just what we see on the outside. They are caused by the electrical brain activity going wrong inside your brain, and that causes the unusual movements, eye rolling and loss of consciousness that we usually associate with epilepsy. But it comes from this underlying dysfunction of the electrical signaling in the brain. So just thinking about how epilepsy is diagnosed, and one thing that I like to tell people when I'm talking about epilepsy is that anybody can have a seizure. So we've already said it's reasonably common, and 1 in 26 of us will develop it at some time in our lives. But actually having a seizure doesn't necessarily mean you'll have epilepsy. So anyone can have a seizure. They can have a very high fever or an illness that causes a seizure. To actually get a diagnosis of epilepsy requires you to have 2 or more seizures at least 24 hours apart. So this is not just a one-off caused by a fever or a disorder. This is a neurological condition of the brain. In order to prove that those seizures are caused by epilepsy, you have a test called an electroencephalogram, or EEG. You'll have lots of other tests as well, but that will be read by a clinical epileptologist, and they will confirm that the abnormal brain activity that they're seeing is the result of seizures. So those two things together, they're having more than one seizure at a reasonable distance apart, and having a confirmation of an electroencephalogram will give you an epilepsy diagnosis. So these are just some slides that I've taken from the World Health Organization, and I just wanted to use them to illustrate a couple of points. The first thing is that there are lots of causes of epilepsy. I'm only going to talk about these ones here, which are the genetic causes of epilepsy. And the other thing I want to point out here is that epilepsy can affect people of all ages, but I'm focusing on epilepsy that starts in early infancy. The impact of epilepsy is huge. There are millions of people on the planet that have epilepsy. Many of them do not have adequate seizure control. But this is really the big impact here. I think a diagnosis of epilepsy comes along with a greater chance of premature death, which is very distressing. So my area of interest is in these very early infantile onset epilepsy conditions that are caused by a change in the genetic code in the child. And these have this rather long and unwieldy name of developmental and epileptic encephalopathies, or DEE for short. And I'm going to call them DEE, obviously, because it'll take me a lot less time to say that. These were first given the name DEE by the International League Against Epilepsy here. And there are two things that these things have in common. So they can be caused by lots of mutations in lots of different genes, but they're all clinically characterized by early onset, drug-resistant and frequent seizures. So the children have a lot of seizures. They start usually within 3 to 8 months of life, and they're very, very difficult to control with drugs. Along with that, there's an underlying pathology of the brain, which is called an encephalopathy. So encephalo is just brain, and pathy comes from pathological or disease. So that will cause a delay, stalling, or regression of developmental skills. So once these children begin to have seizures, they can either stall their development, it slows down, or it can, in some cases, even go backwards. So these two things together, the seizures very early on and the underlying developmental condition, make it ideal for someone like me to come in and try and understand how that's happening. Clinical geneticists have now found 117 different genes that have a change in them which cause a DEE. And each DEE comes with a number. You'll see some of those numbers in my slides. And we're up to 117 as of just now. So that's a lot of different genes that are affected. Humans have about 20,000 genes. So there's a large number of different genes that are affected in this disorder. So it's a complicated scenario. It's not like something like cystic fibrosis, where one gene causes all of the cases. It does come along with a high rate of mortality. Around 25% of children don't make it to their 20th birthday. And that is both a result of the seizures themselves and also of the underlying disorder. And there are currently no effective therapies. It's essentially guesswork for the clinicians to try to stop these seizures and control them, and they morph throughout the child's life as well. So in order to attack a big problem, you need a great team, and this is our great team funded by the Neurological Foundation. Thank you very much. And from left to right, this is me. This one here is Shalokna Banerjee. She's our postdoc on the current Neurological Foundation grant, and she will be making the genetically modified models that we're studying. Next to her is Alex Matthews. Alex is an undergraduate student currently at the University of Otago, and he's interested in developing smart methods to characterize the behaviors of our tadpoles, which will help us to understand a little bit more about whether drug treatments are suitable for use in these models. I'll come back to that later. The two chaps on the end here, these lovely people, are my colleagues in the Department of Zoology. They're both neuroscientists. This is Paul Schuska, and this is Bart Gerten, and they are helping me out with lots of things that I don't know anything about. Because as Sarah said in her Whakatauki at the beginning, teamwork and group, so it's people coming together to solve problems. So let me introduce you to my son. This is Owen. He's my middle child. I have 3 boys. Owen was born in 2003. And the first thing that we noticed about Owen, I think, was that he wasn't very good at fixing and focusing. So he didn't have very good eyesight. And by about 11 weeks of age, he'd been diagnosed with a condition called microcephaly, which is where the brain doesn't grow at the normal rate after the child is born. So that was picked up quite early. He then started to have seizures. So seizures from 5 months. And then this thing that I'll talk about in a minute called infantile spasms came after that. And then a very long time afterwards, because now we have all this genetic information and we can make connections between children right across the planet that have mutations in the same gene and the same clinical symptoms. He was eventually diagnosed with this DEE65 caused by a mutation in this gene here, CFIP2. And that was done by Professor Lynette Sadlier, who many of you will know, who's at the University of Otago in Wellington and runs a clinical epilepsy group there. Just down under here is a list of some of the drugs that my son has had over the years. This isn't even complete. He's currently on phenytoin, which is controlling his seizures for now, but that's a relatively recent addition, and some of the procedures and diagnoses that he's collected along the way. So you can see that these are quite complicated kinds of disorders, and they all have their own little quirks, but they're all characterized by these very early onset seizures. So what I want to do is show you what these infantile spasm seizures look like. So if you do happen to see them in a child, you'll know that this is a medical emergency and that a doctor needs to see these kids very, very quickly. So in this video, which is has been produced by infantilespasms.org, which is down here, you'll see a group of children having very, very subtle movements of the arms and legs that will look to you abnormal, but perhaps not what you think of when you think of a seizure. So I'm going to play that now. You see these babies will suddenly flex their arms and legs. And sometimes their eyes go quite starey, like this one here. And if you see clusters of that sort of behavior, then that is characteristic of these infantile spasms. And this is often how DEEs first present in the clinic. This one is even more subtle. He's just nodding his head, but you can tell that there's a kind of a switch off in the brain there momentarily, and he looks very confused. Okay, so what they tell parents if they see behavior like this in their children is to video it, which is why we have all these wonderful videos of different children that are having spasms. Video it, take it to your doctor, and explain to them what infantile spasms are until you get the response you want. Getting those seizures under control early is absolutely critical in making sure that your child has the best possible start. Okay. So credit that to the infantilespasms.org. I'll just let the video finish. Okay. So, okay, back to something a little bit more strange. So I work on these South African frogs, and they have the wonderful name of Xenopus, which means strange foot. And so Xenopus have been in the labs for probably since the 1960s. They were the first animals to be cloned, in fact, and they've won a Nobel Prize for it, or at least the professor that did it has won a Nobel Prize for it. And what they do is they produce eggs all year round, and their eggs are really large. So female frog here, adult female frog, weighs about 70 grams, and she can lay thousands of eggs in one day. And these eggs are about 1.4 millimeters across, so they're quite large and quite easy to work with. And of course, they develop outside of the body, so you can follow them all the way through development. So here's the first stages of development. These are fertilized Xenopus eggs. You'll see they have a dark half and a light half. The first thing that they do after fertilization is they all rotate, so the dark half is uppermost. And that yellow dot in the center is the female nucleus. It'll come together with the sperm nucleus, and all of the DNA will then be copied and then apportioned out into two cells. So we see the cell divide into two. There it goes. Hopefully you can see this. And then about 15 minutes later, it divides into four. So that takes about 2 hours in real time. That's filmed in time lapse. And the embryo doesn't get any bigger. It just divides up into smaller and smaller cells. And it will do this until it gets to about 1,000 cells. And then something different happens. Rather than just copying the DNA and dividing the cells up, the cells will start to move, and they're still dividing at the same time. And as the cells start to move, you'll start to see structures and organs and tissues appearing within these embryos. So I'm just going to run this time lapse here. You'll see the embryo kind of turns itself inside out. So it's moving, cells are moving inside. This is going to become the back end of the embryo, and this will be the front. And this tissue on top here is going to make the nervous system. So initially it's flat, it rolls up into a tube, seals itself up nicely, and here is the spinal cord forming, and this part will be the brain. So that's how it's built. It starts out as a simple tube that rolls up on the back of the embryo. Same in us as it is in frogs. We don't look quite like this, but that's the same process that's happening. So that took about 24 hours to get to this stage. And then by about 5 to 7 days, we've got a very nice independent swimming tadpole. Okay, so we can make lots of these because these animals have lots of babies, and we can make lots and lots of these tadpoles here. The great thing about these tadpoles is because there's no skull developed yet, it's too early, they haven't developed any bones at all, you can see right through into the brain. And the brain, although it looks very different to ours, is built the same way. So here you can see in a human embryo at 1 month, you've got your tube central nervous system, and here is the forebrain at the front of that tube. Here's the midbrain and the hindbrain and the spinal cord. And over time, those will develop into structures that you might be more familiar with. So the very front of the forebrain here, the telencephalon, develops into the cerebral cortex. So that big lumpy pink thing that we think about when we think about a human brain, that's all built from the very front part of this tube. In a tadpole, it looks a little bit different. So you're looking down onto the top of the head of a tadpole now. Here are its eyes on either side, and nostrils here, and mouth. And this is the brain here. So we've got the forebrain at the front, midbrain, and then the hindbrain that you can't really see here, and then the spinal cord running down the animal. So we've got a beautiful, accessible system that develops outside of the mother in a simple salt solution and has this accessible, visible brain that we can videotape and look at. So the first thing we did was to develop a method of videotaping the behavior of the tadpoles. So we've built several rigs over the years. This is the current one that we're using, and it's built on Raspberry Pi computers, driving Raspberry Pi cameras. And here are our tadpoles arrayed in a 24-well plate, and they're being filmed from above by these cameras and lit from the bottom by a nice bright LED light. And so we can record these tadpoles for about an hour at very high resolution, about 50 frames per second, because they move quite quickly. And then we can track their behavior and see how what we've done to the genetics or what we've treated them with changes that behavior. So we started out with our first Neurological Foundation funding with this chap here. This is Sandesh Panthy, who's since left the group. And he developed a method of tracking these animals using a software called TopScan. And this just basically takes one point in the center of the tadpole, which you can see is nice and dark, and easy for the camera to detect. And it tracks its movement over time. And then it can make some assumptions about that movement. So it can measure the speed and the direction and things like that, but not everything is captured. So these things down here are traces. So if you imagine the tadpole as drawing it with a crayon as it moves around, over the time of the video, we see this trace appearing, and that's the movement track of the tadpole. Controls haven't been treated with anything. These tadpoles here have been treated with an anti-seizure medication called valproic acid or Epilim, which is commonly prescribed in children. And these tadpoles here have had the opposite. So they have been treated with a drug that induces seizures. So as well as drugs that can stop seizures, we have drugs that can start seizures and can keep seizures going indefinitely. And they're quite good for developing these methods before you start turning genes on and off. You can use these to benchmark the behavior. So you can see these tadpoles are moving around a lot more. They've been very busy. They're moving a lot faster. But if we add back in some Epilim, some valproic acid, that is slowing down. So you can see that those seizures are being actively blocked by the anti-seizure medication, which is quite nice. So Sandesh was able to determine several different types of movement that we associate with seizures using these drug-induced models. And this is the one that we'll be focusing on, is the C-shaped contractions. This is a very strong bend of the tadpole that we wouldn't ordinarily see. The tail comes right the way around to the mouth almost. And it sort of resembles those extreme movements that we see during seizures in the kids as well. Very strong contraction of the muscles. These graphs down here just show that if you collect a lot of tadpoles together that have been treated in the same way, you will see that these ones that have had seizures induced behave very differently. They have a different statistical letter up here, which tells you that it's different from the others, both in velocity and in the amount of time they spend darting around, moving very quickly, making lots of tight turns. Okay, and we published this work in the Journal of Neurochemistry in 2023. Before I move on to the next slide, just one thing I forgot to say is because these tadpoles develop outside of the mother and they're in a very simple salt solution in these little wells, it's very, very easy to add drugs to the medium to either induce or prevent seizures. So keep that in mind. That's one of the reasons we use the model. Then I introduced Shalogna down here. So Shalogna originally came over from the UK as a PhD student, and she was instrumental in setting up a lot of our genetic epilepsy work and is now working as a postdoc on our current Neurological Foundation grant. So it's great to have her back on that. We had found a paper that had been published from another group in the United States where they had found 2 children with mutations, changes to the genetic sequence in a gene called NeuroD2. And this had been found to cause seizures. So these children had seizures very much like my son and very much like other children with DEE. And they had started very early in life with those seizures. When they looked at where the mutations were in these children, this is just a way of looking at genes that code for proteins. This is a model of the protein that's just linear. It's not how it actually looks because it would be all folded up. The 2 mutations are affecting this region here. So one child had this one, another child had this one, but they both had the same seizures. So putting that together, the researchers modeled this in Xenopus, which is the exact same organism I work in, and they showed that they could induce seizures by breaking this gene in frogs. So that was the very first time anyone had tried to do genetic epilepsy modeling in frogs, and it's probably the last time anyone has done it as well. So we've been building on that model to try and develop new models of DEEs as well for other genes. But we did a little bit more characterization of this one first, or rather Shalogna did. This is our video tracking. So we have control tadpoles that don't move very much, and tadpoles that have been CRISPR-Cas edited to break the NeuroD2 gene move a lot more. Okay, so they're presumably having more seizures, they're moving faster, they're generally more agitated in their behavior. When we look frame by frame, we can see these are consecutive frames taken from one of our videos of a tadpole, and everywhere where there's a red box around, that we think is a seizure type movement. So you can see this one here has a very tight bend. This one here has a quite tight bend, and so on. So we see these sporadic seizures occurring in these animals. These haven't had any drugs applied to them. They've just had one gene broken, and that gene is causing them to have spontaneous seizures, just like the children with DEE72. But just seeing seizures isn't enough to get you a diagnosis, right? Even if you have more than 2, you still need an electroencephalogram. And that's very, very difficult to do in tadpoles because they're quite small and sticking electrodes on their head would be quite challenging. So we do it a little bit differently. And we do this in collaboration with Paul Schuska in my department. We use a chemical that lights up gives off fluorescent light when neurons are active. And we can just inject this as messenger RNA into our embryos along with our CRISPR-Cas reagents. And we can see neurons actually firing in live tadpole brains. So I've got a video to show you, and then I can show you what it means in a minute. So here is the tadpole's brain. Okay, we're looking down on that. And when you see light, the black background is no activity, and the light is neuronal activity. So that's happening in live. And this is time lapse again. So this is 30 minutes. I'm not going to make you watch 30 minutes worth of video. This is collapsed down into quite a short time. And I think this is perfect for bonfire night because we're going to see Guy Fawkes night, sorry, you're going to see fireworks going off. Normal brain activity looks like Christmas lights twinkling. Seizure brain activity looks like big explosions of light. And there's a really big one coming there. There we go. Look at that one. So that's a seizure happening sped up. And then there's one more towards the end of the video. There it is. Okay. So if we take individual frames from that video and I draw on the parts of the brain, here's the forebrain and here's the midbrain. And just like our brain, it's divided into 2 hemispheres, a left and a right. So you've got those illustrated here as well. And these times here just represent minutes. So after 0.6 of a minute on the video, we saw a starting of some kind of strong electrical signal just on one side of the midbrain. After a few more minutes, this spreads to the second hemisphere. So it's moving across from this focal start point into the other side of the midbrain, and then gradually over time strengthening and moving into the forebrain. So our DEE75 tadpoles are indeed having neurological seizures, and we can track these peaks of activity on a graph here with time. So those 3 peaks that you saw are clearly shown on this graph, and this is where it ended up here. So we're able to show that the neuronal activity looks exactly like we would expect for an animal that is having an epileptic seizure. So now we should be able to take any one of those 117 genes, break them in tadpoles, and do exactly the same kind of thing. Obviously, don't have time to show you all of those. What else can we do with them? Well, Shalogna was very keen on drug repurposing, and she did a lot of reading around for her PhD. And because our aquatic models are great for drug testing, because you just have to place the drug into the wells with the tadpoles and then see what happens, she thought this was a really good idea. Now, normally anti-epileptic medications are really anti-seizure. So what they do is they dampen down the electrical activity of the brain, but they do it quite indiscriminately, and they are not without side effects. So you might have side effects that affect other parts of the body, but you might also have a general dampening down of electrical activity, which is not ideal. So the idea that Shalogna had is maybe you can look at inflammation instead. And so she thought that picking some drugs that targeted inflammation that were already being prescribed for other purposes and testing those would be a really good idea. And she was absolutely right, because one of the drugs that she selected was called losartan, and that is commonly prescribed to lower blood pressure in people with hypertension. We think it works via anti-inflammatory action, and we know it works like that in the blood vessels, but we think it also works like that in the brain. And there's been at least one study of patients that are on losartan for their blood pressure compared to patients that are not, and whether they develop seizures later in life. So that seems to protect against developing seizures in people with hypertension. In our tadpole model, what Shalogna did was very painstakingly count all those little C-shaped bend contractions in the tadpoles before treating with losartan and after treating with losartan in our NeuroD2 model. So these filled in purple triangles that are pointing upwards are the baseline. These are the tadpoles having their baseline seizures. And then we have the open down pointing triangles, which are afterwards. So about 20 minutes after treating, she counted the seizures again. And this is exactly what would happen if you went on a clinical trial for epilepsy. You would have a baseline taken, you would record all your seizures over a certain number of days, then you would start the treatment, and then you would record the number of seizures moving on, and hopefully that goes down if the treatment is effective. So these are the same tadpoles. This tadpole here reduced its seizures from around 70 down to around 25. And you can see that most of the tadpoles reduced their seizures by some degree in this medication. So this is not an anti-seizure medication. This is something completely different. 16 out of our 18 tadpoles in this study had fewer seizures, and the average number of seizures over the whole group was about 4.7% reduced. And we published this in our paper in Genetics in 2024. However, what Shalogna had to do to get this data was many, 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 many horrible hours of counting frames from videos. So we have 24 tadpoles in a plate. We film at 50 frames per second. So that's 3,000 frames times 24 per minute of video. And then she has to do it after drug treatment as well. The other issue that we have is that anything that involves manual counting is subject to unconscious bias. So you have to try and remove all of that in order to get really nice data for your research. So what we have Alex Matthews doing, along with Bart Gerten, is to come up with smart ways to measure these behaviors and quantify them in our tadpoles. I told you before that when we track our tadpoles, we just have a single dot in the center of the tadpole, and we follow that movement around using TopScan. The idea of this type of detection is that we will train machine learning algorithm to label the tadpole for us. So we went through a number of frames and we told the computer where the left eye is, where the right eye is, where the base of the stomach is, and then 5 points. I know it's only 3 on here, but 5 points along the tail. And we did that for a number of frames, and then we allowed the machine to learn from that. And now it can accurately add these points to the tadpoles frame by frame. I wouldn't say quickly, it does take quite a long time, but then all of that data goes into a clustering algorithm written by Alex and Bart, and then we get out different types of behavior. So not only our C starts or our C contractions, but lots of other behaviors as well. And we're hoping that we'll be able to use this in our models to really accurately count the number of seizure events, and also to see the differences between the different genetic models that we've made. And this is my last slide, but what we're trying to do is collect lots of these different models. There are 117 genes to choose from. We have picked genes that we think might be involved in synapses. So these are the little connections between neurons where signals are released from one and accepted by another. So not all of the wiring in the brain is continuous. You have these little gaps called synapses. And so we are targeting those particularly because we think that they might have a common cause that means that when you break them, you end up with a very similar clinical phenotype of early onset seizures. So these are the 4 models that we're working on so far. You see they've all got a DEE and a different number. And the second thing in brackets is the name of the gene. And then after that, we've got the name of the person that's responsible for making those models. We've got various students involved in it. And what we're going to do is to characterize those, find the commonalities in the behavior, but also to look at how their brains are changed by looking at the way that all of the genes in the brain are up or down regulated. So every cell in your body has a complete set of the DNA, but in every cell, you will have different genes that turn on and off. So we're thinking that in our DEE patients, what we might see is the same genes being turned up or down, and we can use the power of having multiple models to try and pick the ones that are important, to try and pull out the signals from the noise or find the order in the chaos, if you like. So far, we've only done this with NeuroD2, and our preliminary results are showing all of these little dots in purple represent a gene that has been turned up significantly in a DEE 72 tadpole brain, and all of these sort of teal colored ones have gone down. So that's too many genes to chase up. So we're hoping that with the power of multiple models and comparisons, we'll be able to pull out genes that are really, really critical in this process. And that might tell us where to target our drug treatments, because obviously there's lots of drugs out there that we could test. We don't have time to do everything, so we're trying to narrow that down. And that's about all I have time for. So hopefully I didn't overrun too much, Sarah. And I'm happy to take questions.
That was great. Thanks, Caroline. And I guess the obvious question is, what is the next step? So if you find a drug like the blood pressure one that looks like it could be effective for certain types of seizures, how would you get that from the tadpoles to the babies?
Yeah, no, I probably wouldn't start with the babies, to be fair. That I think we would not be doing that ourselves. We're modelers, we work with model organisms, and so we would be trying to attract the attention of clinicians at that stage and hope to pull together enough evidence for a clinical trial. So the good thing about repurposing drugs is these are drugs that are already considered to be safe. They've already gone through testing processes, possibly not in small children. So they've ticked some of the boxes already, and hopefully wouldn't be too onerous to start trialing them. And I guess it's have failed a lot of medications that parents might be willing to try something that is a little bit more off the label.
Yeah, that's true. Could you tell us a little bit about what these seizures are actually doing to the development of the children?
Yeah, so it's not the same in every child, and it even seems to be different between different children who have the exact same typographical error in their DNA. So I think a lot of it is timing and recognition of the condition early on and getting treatment started early, finding a treatment that works. So in terms of what happened with my son, he was able to sit up and interact with toys at one point in his life. Then he developed some rather nasty seizures at about age 8 and was unable to do those things afterwards. So they can have developmental regressions even quite later on in life. But the stalling really starts to be seen with these kids. Don't tend to sit up, they don't tend to crawl, and everything takes a little bit longer, if it's ever achieved at all. So in the case of my son, he's a wheelchair user, he won't walk.
And does the research also have potential for people that do research into adult epilepsy? Can it provide some guidance to those teams as well?
Definitely with the losartan, because this retrospective study that was done in German patients that's been published showed that losartan was protective against seizures. It's a correlative study, but everything is a correlative study at the beginning. So now what they would need to do is deliberately start treating people with losartan and seeing how they track through time.
I picked up how you said that we can see the data now because we're collecting a lot of data from medications that people are on, and then we can see if those people also go on to have seizures later in life or not. So is there a lot of things that people can do at just looking through those records now of people who are on different medications and what ones might be potentially preventative for epilepsy?
Yeah, I think there's probably a lot of data out there that can be mined by clinicians. The problem is that clinicians are already very busy treating patients. So they, the people that you want to target, or the people that will look into this, are clinicians who are also researchers and can employ people, perhaps funded by the Neurological Foundation or similar, to do that sort of looking through the data and making sense of that clinical data and correlating it with the drug treatments.
Yeah, we have funded some projects to get a registry underway for sudden death in epilepsy as well. So it's great to be able to contribute to some of the research that's going on, and obviously a lot more is needed to be done for DEE. I'll just ask a question that's come through from the audience. With the prevalence of childhood epilepsy, do some children see reduced symptoms as they age, or is that different types of epilepsy that isn't genetic?
Yeah, so there are some types of epilepsy that they may or may not be genetic that do resolve with time. So juvenile myoclonic epilepsy would be one of them. It's expected to resolve with time, and as adults, they don't have any symptoms. With DEE, there's no evidence that it disappears. What does seem to happen is that the kids go through a sort of golden era in their early years after this horrible onset of infantile seizures. They can sometimes have a period of 3 to 4 years where they don't have any seizures. And that may be on medication. But then when they hit their older years, and particularly in puberty, they tend to suffer from more and different kinds of seizures. And I really, I don't speak to that many parents of people with children with DEE, but I'm in a group on Facebook of collected parents with kids with the same gene that my son has, and they're all at different ages and stages. And of course, if they're born now, they're immediately genome sequenced and immediately diagnosed. But actually, the treatment hasn't changed. So although we've got a name for it and we've got a little group on Facebook, we still don't have any kind of change in the way that it's treated. It's exactly the same. And I think that's what we want to see change in the next 10 years or so.
Okay, we've had a couple more questions come through. Kia ora, Lisa. So Lisa's part of a charity for tuberous sclerosis complex.
Oh, yeah.
Which is a genetic condition associated with epilepsy.
Mm-hmm.
Do you know how DEE differs from TSC, or is it very similar?
It does differ. It does present, I believe, the same way. So that was something that was ruled out in my son very early on. I don't know a huge amount about it, but there's a thing called a Wood's lamp test, which they can use to detect patches in the skin, I believe. I think they do have infantile spasms, but I believe that the prognosis is a lot better for tuberous sclerosis than it is for DEE caused by genetic mutations. And again, I mean, they will be caused by genetic mutations as well, I imagine. But yeah, I think they do diverge. They have that initial clinical similarity, but then they diverge.
Okay. So we've got an interesting question about programs in kindergarten that might help teachers spot seizures. Do you know if early childhood educators are being educated themselves to spot these type of seizures?
I couldn't tell you whether they can specifically spot infantile spasms, but I know that, like, particularly in Dunedin, we have an epilepsy nurse who goes around to schools and kindergartens and educates the teachers in what to look for. So if a child does have its first seizure at kindergarten, then they would at least know how to deal with it. So that's on top of the first aid training that they would also have as well. I've never seen infantile spasms covered in first aid training. It might be, but it's unlikely that you would find an infant just abandoned in the street having seizures, I guess.
Yeah, no, I don't remember that from any first aid training that I've done.
No.
How does it relate to the petite grand type seizures and how those might progress or be affected by drugs?
Yeah, so the terminology has changed a little bit, and it's focused more on whether the seizure has a general or a focal onset, which can help, obviously, with diagnosis. So if there's a defect in just a small part of the brain that might be a good candidate for surgery, then you might expect to see a very focal onset seizure that starts always in the same place. These tend to be more general, at least from what we see in the tadpoles. I can't read EEGs myself. I have no idea what those squiggles mean. But so we don't tend to use grand mal and petite mal seizure terminology anymore. But essentially, my understanding of that is a grand mal seizure would be called a tonic-clonic seizure. There would be a period of muscle contraction followed by convulsions and probably loss of consciousness. Whereas a petite mal seizure is a loss of consciousness, and children tend to drop to the floor. So I haven't experienced any of those, but my son has never been in a position where he's been standing independently. So we would not know if he was having those kinds of seizures. And I imagine it's the same for a lot of these kids. We only see the big, impressive ones because we probably miss a lot.
Right. So could you replicate? My questions are bouncing around here. Oh, I think it's just gone, that one. Oh no, here it is. Could you replicate a person's genetic differences using CRISPR as an aim to look at treatments that way?
Yeah, so we're starting with the low-hanging fruit by just breaking the genes, because essentially with about 60% of the genetic DEE, that is the cause, that the gene is just broken. And so it doesn't make a functional protein and it doesn't do its job properly. In the case of my son's gene, it's not that simple. We have to actually replicate the mutation, and that's taking a lot longer. We're trying to do that with messenger RNA. We could do that with CRISPR, repairing with a template that matches the change sequence. So yes, it's just another step in the process. So as we get more confident and as this takes off, hopefully as a method that's being used to study them, then we will have more resources to do that. So it's possible, but it wasn't the obvious place to start.
The same person is also asking about whether organoids have been used in these models yet.
Yes, they are. So I'm in touch with at least one group over in Brazil who's using organoids to study my son's mutation. They've taken cells from a different child with the exact same mutation, turn those into induced pluripotent stem cells, and then grow those into brain organoids. So yes, it's being done. And there may come a time when we don't need to use animal models at all, because organoids will get better and more representative of what's going on. So I think at the moment, using cells, using organoids, using animal models all needs to work together, but we might be slowly moving away from animal models in the future. But yeah, definitely organoids are being used. Obviously, you won't see movement because the brain doesn't move by itself, but you can see everything.
Yeah. So what might the pathway from your research to clinical treatments available in the future look like if somebody else took on the work that you've started and took it towards a pathway to clinical treatment? What would that look like and how long might it take?
Yeah, I guess it depends on whether this is a repurposed drug, because if it's a repurposed drug, it might be quite quick. And it depends who's running the trial and what the parents are prepared to agree to. So I know that Lynette Sadler is looking at N=1 treatments for individual genetic epilepsies, and I think that's probably being done in other parts of the world as well. And those would be things like antisense oligos that are designed specifically for one person. But the pathway, it obviously can be very slow. If you think about how long it's taken to get the cystic fibrosis drugs into New Zealand and available to the kids with cystic fibrosis, it's taken a very, very long time to do that. So it's possibly not around the corner.
Is it more of a personalized treatment, do you think, where individual treatments are going to be used for individual patients?
I think that's already happening, and certainly in other parts of the world, and I think it will here too. But what we're hoping is that there might be something that is more suitable than an anti-seizure medication that can at least help. So if it turns out that anti-inflammatory treatment that is otherwise harmless could be used in our kids, and it changes the trajectory of their development a little bit, it might give time for these other drugs to be developed.
So if a parent has just had a diagnosis, what support is available for them? And I guess this is something that you know very well.
Yeah, I guess you're going to turn to places like the Epilepsy Foundation and online. And to me, the greatest support has come now from parent groups on Facebook and other websites. So there are websites that you can sign up to and that you can share information in private rooms with other families with the same condition. And so a lot of it is anecdotal, parent to parent. And of course, with things like Zoom, we can meet these parents. We actually had a meeting of parents from across the world with my son's mutation. And we had Zoom translating people from all across the world together. And we were talking as if we were all talking the same language. It's fantastic. And yeah, so it's very difficult for a GP to give support because they will not have seen anything like it before. And so when we came into this process, there was very little because we didn't even know what to call it at the time. It was just microcephaly and seizures. And we were just under general pediatrician, no neurology input really at all. So yeah, I think support can be everywhere, and it's just worth putting your feelers out everywhere.
What kind of support is available from Epilepsy New Zealand?
I'm not aware of any, but I'm sure there is. And they would certainly be a first point of contact. I think they would have information about DEE on their website. I haven't personally looked though.
So potentially resources and references to support groups. They may have that sort of information.
Yeah, I would hope so. But again, sorry, I haven't looked.
So you mentioned at the beginning that there was not that much research going into DEE. So what is going on around the world that you know of, and why is there not a lot of it?
I think there is a lot, but it's bigger than that. So it's really taken off in the last few years, and there are lots of groups that are pulling in funding to work on DEE. We have focused on genes that we know there is at least one patient in New Zealand, and we've had that information shared with us, which is great. We don't know the identity of the people, but we know that those genes have at least one patient in New Zealand. So I think it's difficult for us to do much on a bigger scale from a small country, but there is lots of DEE research going on across the globe, and they'll be using very different approaches as well.
So I think we have managed to answer all the questions tonight, which is fantastic. Thank you for keeping your talk to a length that we had time to answer everybody's questions and give people that valuable feedback. And thank you also for a very informative talk from a personal level and also from a scientific level. I'm sure it's been really helpful for people, and I just want to really say thank you for joining us tonight.
Well, thank you, Sarah, and thank you very much for inviting me to give the talk, and thank you to everybody for showing up and listening. It's been a lot of fun. I hope I did a good job of answering your questions. Thank you.
We've been having some comments coming through saying very helpful and informative, so that's great.
There is a hand up, Sarah. I don't know if that...
I think I'll let our IT deal with that. But yeah, thank you very much for everyone for joining us tonight as well for this very informative talk. And it will be available on our website soon. And I will now end the meeting and wish you all a very good night.